I don't understand. The baby became
Okay, to begin. I can tell it's exam day. The exam, of course, is this evening. We'll see you all again at 5. Check the home page to see what room you're supposed to go to. You're being divided up to different rooms according to the first letter of your last name. All that's listed on the BMS 300 homepage. Today what we're going to do is going to start a whole new section. Today's new section is going to be on the cardiovascular system. And so what we're going to spend our time on is basically the mammalian heart, its structure and function. And we're going to talk about its chambers and vessels and valves and the blood flow through the chambers. But as we move through this, in order to give you some idea of why to bother to have a heart, I want to give you some sort of comparative physiology, and just a little bit. And we'll talk about the idea of very simple organisms, a single-celled organism or an organism like a sponge or a coelenterates. And the main idea here, the main function of that is going to be that when you're a single-celled organism, or even if you're only composed of a couple layers of tissue, you don't have to worry about a circulatory system. You may move fluid through the chamber if you're a jellyfish, but in fact you don't have much problem with oxygen and nutrient delivery, because you just take the oxygen and nutrients out of the world around you, and you get rid of your waste product organism. Once you have structures, and they started as this two-chambered structure, where there was an atrium which receives deoxygenated blood, and then a ventricle that pumps blood to both the gills and the whole systemic vasculature. And I'll make a brief and a sort of crude drawing of this, trying to show that there's one pump in a fish, and that pump pumps the blood first to the gills, but the very same pump is forcing that blood after it moves through the gills to the rest of the vasculature. So it's basically one pump to serve the whole system. Apparently that worked just fine as long as creatures were living in water, but once they came out on land, then it required a more complicated heart. And in both cases, there are 4 chambers. And in both cases, what we see is that these 4 chambers are organized as 2 pumps in series. One pump is dedicated to moving the system. Well, to understand how blood flows through this, I'm going to do this kind of thought experiment. I'm going to simplify all of this down to what I call a 4-box model. And we'll talk about the 4-box model as being the right atrium and the right ventricle, the left atrium and the left ventricle. The right atrium and the right ventricle will have have tubes flowing into them and out of them. The right atrium will receive deoxygenated blood from something known as the vena cava. The vena cava is a joining of the superior and inferior vena cava. This is blood returning to the heart. This will bring us to a kind of nomenclature. It tells us that when we talk about vessels bringing blood to the heart, carrying blood toward the heart, that will be a vein. So these will be the vena cava in here. And then on the right side, we'll see the blood is going to flow from the atrium to the ventricle, and then the ventricle is going to move the blood to the lungs. It's going to move the blood to the lungs through a tube called the pulmonary artery. And again, this is going to be the nomenclature. An artery is a tube carrying blood away from the heart. Now the left side, it's the same organization. Again, we have an atrium and a ventricle. The left atrium is going to be receiving oxygenated blood, returning to the heart through those pulmonary arteries. The pulmonary arteries are carrying blood toward the heart, so those are arteries. Now we'll see that the blood comes through the atrium, into the ventricle, and then leaves the ventricle in a structure called the aorta. Now we're going to see there are a series of valves here, and we'll walk our way through the valves. First we'll describe the valves in the 4-box model, and then we'll put them actually into a structure that looks more like a heart. But there are going to be a set of valves between the atria and the ventricles. Those are atrioventricular valves, and that's the nomenclature I'll use, is atrioventricular, right or left atrioventricular valve. And I prefer that nomenclature because it tells us where it is. But in fact, that nomenclature isn't universal. Sometimes you'll see these referred to as a tricuspid valve or a bicuspid valve. Sometimes referred to as the mitral valve. And we'll work our way through these and we'll talk about them. But most of the time we're going to talk about atrioventricular valves. And then there are going to be pulmonary valves. There's going to be a pulmonary valve. This is going to be a valve that's to the opening of the pulmonary artery. It's going to be on the right side. And then there's going to be a valve called the aortic valve, which is on the left side, which is found in the opening to the aorta. And we'll walk our way through that. But let's first just think about this a little bit and take one of these panels and simply label it as why we need a heart. Basically, why have a heart? And if you're a single-celled organism, if we go back 1.5 billion or 2 billion years, and everything was a single-celled organism, even if it was a eukaryote, really didn't need some sort of a pumping system because this lives in the ocean. And so oxygen can easily diffuse across the phospholipid bilayer. Nutrients can easily diffuse across. So diffusion in and out of the cell is just fine for oxygen and nutrients. What's more, though, is if it's a eukaryotic organism, if it's using aerobic respiration, it's going to generate carbon dioxide as a waste product. Again, carbon dioxide easily moves from the inside of the cell to the out. We don't need a pump. The other waste product we'll see is ammonia, NH3. We'll talk more about ammonia when we get to the kidney. But the ammonia is a byproduct of the breakdown of protein. Anytime we digest proteins, anytime we metabolize proteins in cells, we end up with a byproduct of ammonia. It's really toxic, but it's pretty water soluble, so it easily diffuses out. Even if we imagine jellyfish, the so-called coelenterids, they're basically 2 layers of cells. There's an ectoderm, comes around sort of a lip. And then on the inside, there's a structure called the gastroderm. And in between, there's this structure called the mesoglia. That's the jelly of the jellyfish. All I want to point out here is that if you've ever watched a jellyfish function, you know it has this bell that opens and closes and opens and closes. And what it's doing basically is it's moving water into the bell and back out again. It's circulating the water through the system, but it doesn't need an intrinsic pump. It doesn't need an intrinsic pump because both the gastroderm, where nutrients can diffuse in, and where oxygen can diffuse in, or from the outside, oxygen can diffuse in, these cell layers are so thin that they're never more than 1 millimeter or so away from the surrounding water. So that means we can simply use diffusion across distances of 1 to 2 millimeters. We can use the diffusion across those short distances both to obtain oxygen and nutrients, as well as to get rid of ammonia and carbon dioxide. But no intrinsic pump is required. When you begin to look at 3-dimensional organisms, even if you're looking at arthropods, we begin now to need some kind of a circulatory system so that oxygen and nutrients can be delivered to various parts of the organism. The one I'm going to tell you about, of course, is the teleost fish. This is like a goldfish. Story could probably be told for shark too, but the way I want to draw this is one of my terribly primitive fishes. So there's the dorsal fin. Make that a little bigger. Ventral fin. And up here's the head. There's an eye. And then there's an opening here. And the opening allows water to move through. And if you've ever watched a goldfish, you know there's a bony structure back here called an operculum. The operculum is that bony thing that when you watch a fish in a fish tank, that's what's moving in and out. What the fish is doing, it's hard for you to see from that, but what the fish is doing is it's bringing water in through its mouth. And then the water is leaving at this site when the operculum moves in and out. But if you look beneath the operculum, there we find the gills. And the gills are the oxygenating organ. Then the structure we're interested in today is the structure back here. This is what we're going to call the fish heart. The fish heart has two chambers. It's an atrium and a ventricle. The atrium is a receiving area. And the atrium is receiving blood. It's receiving deoxygenated from the tissues. The ventricle is the pump. This is the muscular pump. And it's going to move blood, when it pumps, it's going to move blood in this direction toward the gills. And if we look at the gills more closely, we see that as an oxygenating organ, they're very delicate structures. And if you've ever looked at the fish's gills, you'll recognize their whole series of finger-like structures covered with even tinier extensions. But if we draw them simply here, the general body plan is to have a structure like this. Where out here, this is epidermis, or basically, this is an epithelium. I can orient you with respect to the outside world. That epithelium is going to sit on the basal lamina. And then immediately beneath the basal lamina is going to be a blood vessel. This blood vessel is going to flow in, and it's going to flow out, basically. So the blood flow is going to be in this direction. Put it in here. The blood vessel is lined with a series of cells. This is a term that we're going to use lots and lots. These are the endothelium. And they line the gill capillary. I don't want to belabor this too much, but what I want to point out to you, and I wish I'd made this a little bigger, is that there's this lining of the gill, and the blood is flowing through here. But there are only two cell layers between the water moving across the gill and the blood. The first cell layer is this epithelium. So the oxygen can diffuse across the endothelium and reach the blood. And because the blood is being pumped by that single pump we showed above, the blood is moving through the gills. It comes in deoxygenated. Get that blue. And it leaves oxygenated. That's basically how the gill works. Carbon dioxide is also going to diffuse out of the blood and into the outside world. But from our perspective here, what I want you to see is that the blood flows through the gills, goes into those fine capillaries, and then is collected and goes out to the rest of the vasculature. So this is the blood. I should say O2 rich blood to the remaining systems. So this blood, once it's oxygenated, now goes to the muscles, it goes to the gut, goes to the brain, goes to the liver, goes to all those different places. But there's only one pump. And that pump here is pumping blood first to the gills, through that capillary bed in the gills, and then even though we've lost all that energy, is the blood moving through the gills, there's only one pump to move the blood through the rest of the system. Fish have gotten along just fine this way. But oddly enough, if you look at an invertebrate, a cephalopod invertebrate, a squid, and if you look at the squid solved this problem differently. What the squid does is it has a pump here that pumps blood to the gills, and on the far side of the gills, there's another pump. So it has basically a pump to pump deoxygenated blood to its gills, and then another pump on the far side to pump the blood to the rest of the system. Teleost fish never figured that out. Because we evolved out of this sort of structure. We now, because our ancestors, once we got out on land, had to change the structure. And once on land, we had to generate two separate hearts. One of which is dedicated to pumping blood to the oxygenating organ, in our case the lungs, and the other is used to pump blood to the rest of the vasculature. That's the model of the four-chambered heart. If you talk about amphibians or reptiles, you see they're often three-chambered hearts, and reptiles are often a septum between the two. But for our purposes, what we want to talk about here is we want to spend some time talking about the four-chambered heart. Let me get rid of this. So the four-chambered heart is the one we're going to spend the rest of our time talking about. And it's a modification. If you look at the embryological development of the heart, it develops from basically a two-chambered heart. And there's some elaborate folding that it does to generate the four-chambered heart. If you take an anatomy course or an embryology course, a developmental biology course, you'll spend some time on that. But what I want to do here is just summarize the ideas of the four-chambered mammalian heart. I'm going to simplify this way down for our first discussion here. And what I'm going to show over here on the left-hand side of the board is the four-box model. And we'll give the proper terminology here. But as we work through this, all I'm trying to show with the four-box model is describe the blood flow through the heart. And we're going to talk about these four boxes as being rooms or chambers. We're going to talk about the valves as being like doors. And we're going to simplify this down to something we think about every day. And then we'll move on from that thinking about the proper three-dimensional structure of a mammalian heart. But first, let's talk about the four-box model. Now, the convention is to look at the heart as though you're looking at me sitting out there. So this is going to be right, this is going to be left. You can think of it as looking at a cadaver on a gurney. So it's what you're looking at, and it's from the position of these chambers will be as though you're looking at the heart, looking down on them. So this is going to be left. Sorry. So over here is going to be right. This is going to be left. So the first box I'm going to draw here, I'm going to label this as the right atrium. The term atrium comes from the Latin. It means a receiving area. We use this term in everyday speech. If you go to a really large building, if you go down to Denver and you walk into one of those big skyscrapers in Denver, you often enter this great open area. That's the atrium. It's the receiving area. Well, it's true for the heart too. This is the receiving area. And what we're going to see is where it's coming in. And again, this is not anatomically correct. Just want to walk through the parts. Is this structure called the vena, V-E-N-A, cava, C-A-V-A. The vena cava arises from the fusion of the superior and inferior vena cava. I'm going to move this off to the side because I'm going to need that room. So the superior is above. This is going to be the inferior. But they join to form the vena cava. That enters the right atrium. That's a separate room. Beneath that room, over here, is another room, another chamber. This chamber is going to be the right ventricle. We can think about these boxes as rooms, as three-dimensional rooms. And we can imagine the blood flow is coming this direction. And this is going to be O2, poor blood. Fancy way of saying that is deoxygenated blood. This is deoxygenated blood returning from all the tissues of the body. The deoxygenated blood comes into the right atrium, and then it flows from this room to this room, but in order to get from one room to the next, there's a door or a valve. We call this a valve. This is going to be the right atrioventricular valve. The right atrioventricular valve, there's meaning in that term. It tells you it's on the right-hand side of the heart, right? Atrioventricular tells you it's the valve, it's the door, it's the opening between the right atrium and the right ventricle. This is a separate room. The only pathway for blood to go from this room to this room is through this door. And that door in biology has a name. It's the right atrioventricular valve. Now, in some cases, you may see this referred to slightly differently. There's a common nomenclature called the tricuspid valve. The tricuspid valve, tri meaning three, cuspid meaning two teeth or leaves. The tricuspid valve looks a little like this, where there are these two leaves side by side. There are three of them, and then there's one behind the plane of the blackboard. So there are these three valves. I've shown two leaves here. There'd be a third leaf behind the plane of the blackboard, or the whiteboard. And now the blood's going to flow across that valve. This is a really floppy door. It opens very easily, closes very easily, and hold on to that idea because we're going to come back to it. But the blood flow now is from this room to this room. How does the blood get out of this room? The blood leaves into this structure known as the pulmonary artery. Now, this can sometimes get confusing, but there's an easy way to hold on to it. Whenever you see the term artery, an artery is carrying blood away from the heart. So arteries carry blood away from the heart. Veins, as in the case of the vena cava, inferior and superior vena cava, these are carrying blood toward the heart. So veins carry blood toward the heart, arteries carry it away. Just as there was this valve between the right atrium and the right ventricle, there's also a valve here. This valve has a name. I have to come down here with it. That name is called the pulmonary valve. Sometimes you'll see the pulmonary valve referred to as the right semilunar valve. This is an old name. It was coined probably in the Middle Ages, or sometime in the Renaissance, because when they began dissecting out hearts, looking at gross anatomy, they looked at the valves in the heart and they said, it looks like a half moon, so they called it a semilunar valve. I much prefer the term pulmonary valve because it tells me that it's at the interface between the right ventricular chamber and this tube that's going to the lungs, the pulmonary artery. So that's the pulmonary valve. But the blood flow through here is going to be from the right atrium to the right ventricle, and then from the right ventricle out across that valve. We'll discuss this in more detail, but that valve is much, much stiffer. It's much, much stouter than these atrioventricular valves. The atrioventricular valves are pretty floppy. They're easily opened and closed. This guy is much stiffer. Similarly, on the other side, we can look at the other four boxes, other two boxes rather. This is going to be the left atrium. It's receiving blood. It's receiving blood coming from the lungs. I'm going to show this is a tube, and this is going to be the pulmonary vein. There's the nomenclature again. When you're sending blood away from the heart, that's going to be an artery. When you're bringing blood toward the heart, that's a vein. So this is the pulmonary vein. This is the pulling together of all the blood, the pooling of the blood coming back from the lung. The blood's been pumped to the lung by the right heart. The blood goes through the capillaries in the lung where it's oxygenated. The oxygenated blood now comes back to the left heart through the pulmonary vein. Comes back in this direction, enters the left atrium. Again, this is a chamber, it's a room. Down here, there's another chamber, another room, and these rooms are separate from one another. And this is the left ventricle. More accurately, it's the left ventricular chamber, it's the opening. And just as we saw on the right, we again see this valve. And the valve between the left atrium and the left ventricle, of course, has a name. It's called the left atrioventricular. Left atrioventricular valve. Again, it tells you where it is. It's on the left side. It's between the atrium and the ventricle. I think that's logical naming. But in fact, the left atrioventricular valve has another name. It's called the bicuspid valve. Where the tricuspid valve had 3 leaves, the atrioventricular valve has 2 leaves, and therefore it's called the bicuspid valve. It has another name. It's called the mitral valve. This is commonly used, term mitral valve, but it's pretty arcane. It comes from the idea that when the Renaissance anatomists looked at this and they saw these two leaves, they turned them upside down. And they said it looks like a bishop's mitre. If you've ever seen the Pope all decked out in his regalia for a special occasion, the Pope is the Bishop of Rome. And as the Bishop of Rome, he is basically is over the churches in Rome. And he wears this funky hat at these big occasions, at formal occasions. And this is called a mitre. And so this is called the mitral valve. So basically, then finally, out here, there's another tube that's going to be leaving the left ventricle. And again, this is not to scale or anatomically correct, but this is going to be the aorta. And again, between the room and the tube, there is this valve called the aortic valve. And the flow of blood through the system is to have deoxygenated blood enter the right atrium, flow across the right atrioventricular valve. And we'll see a little later when the muscle in the right atrium when the muscle in the right ventricle contracts, it forces the blood out over the pulmonary valve and into the pulmonary vasculature. The same pattern occurs on the left side. These are two pumps. They're in series. They're in series in such a way the blood comes in the right side, passes through the lungs where it's oxygenated, comes back to the left side. Enters the left atrium, flows across the left atrioventricular valve, and then we'll see a little later when the muscle contracts, that will force open the aortic valve to flow the blood, to move the blood out into the systemic vasculature. The first tube will be the aorta, and then it will branch and branch from there. But that's the blood flow through the system. Let me show you a couple of slides. The first slide is a much better picture of a fish heart than what I can draw on the board. Why'd you do that? And what we're looking at is pretty much a much nicer image than what I had on the board, but it says the same thing. It's telling you that back here, where we have the heart, this is the atrium. The blood flows in here, flows into this muscular structure called the ventricle. When the ventricle contracts, it forces the blood to go into the gill capillaries. The blood's oxygenated in the capillaries, but the very same forces that are driving the blood through the capillaries of the gill also drive that blood through the rest of the systemic vasculature. One pump does all the job. That's not true in humans or in mammals because we had the four chambers. This next picture is one to try to help you remember the idea of the mitral valve. This is a picture taken, it was at the Met Gala several years ago. I think the theme was spirituality or something. And Rihanna decided to go dressed as a bishop. So I think this was actually from the Bishop of Milwaukee. And so this is a bishop's mitre. And this is how the blood would flow through that mitral valve, is that the blood would flow through Rihanna's head down from the atrium into the ventricles. But to help you remember this idea of the mitral valve, which will be on the left side. So this is a four-box model of the system, but what we want to think now about is something that's more anatomically accurate. And we'll begin here, and I'll just put this on the board, and try to make it look more like what you'd think of as a heart, rather than the four boxes. But the four boxes are useful because you can think of each of those chambers as being a room, and you can imagine following the blood from room to room. And now what we want to do is we want to think about how those rooms in the four-box model look more like what we'll see in a biological organization. The names are going to be the same. We'll still have an atrium and a ventricle. We'll still have those appropriate valves. It's just now we're going to round them off a little. So again, on the right side, structure I'm going to draw. This is said to be the right atrium. I'm going to put two lines around here. This is the right atrium. I've drawn two lines because there's going to be a thin layer of muscle called the atrial myocardium. Again, you can suss out the meaning. Myo meaning muscle, cardium being heart. So this is going to be muscle of the heart. The wall of this chamber will be the atrial myocardium. But there's an opening in the center of it, which is the chamber. Coming into the chamber will be the vena cava. Between the chamber of the atrium and the next chamber down here, this is going to be the right ventricle. And the wall here is going to be the ventricular myocardium. We'll see that the thickness of that piece of muscle is much, much bigger than the very thin wall, the thin myocardium of the atrium, but it's still myocardium. Then coming out of the chamber here, of course, is going to be the pulmonary artery. It's carrying blood to the lungs, that's pulmonary. Carrying blood away from the heart and to the lungs is the pulmonary artery. And at the interface between the chamber and this tube, the pulmonary artery, will of course be the pulmonary vein. Well, what am I saying? Pulmonary valve. So this is the pulmonary valve. It's found at the opening of the pulmonary artery. And what we're going to do is we're going to see that the blood flow is going to be from the atrium across the atrioventricular valve. And then we'll see when there's contraction of the myocardium, that's going to generate a force. It's going to open the pulmonary valve and send blood out into the pulmonary artery. Yes?
So is that blood deoxygenated blood that's coming into the heart and then leaving via the pulmonary artery to be oxygenated by the lungs?
So let's follow the path. We have deoxygenated blood, or oxygen-poor blood, coming into the atrium, coming across the atrioventricular valve. It's still deoxygenated. Comes into the, now this is really the main pump of the system, is the ventricle. When the myocardium of the ventricle contracts, it's going to generate a force on the fluid inside the chamber. That force is going to open the pulmonary valve. The pulmonary valve is now open to this tube called the pulmonary artery. That's one tube. That tube is now going to go to both lungs. It's going to branch and then to go to the lungs, and then it's going to further branch and branch and branch and branch to get to tiny capillaries. And the capillary will be composed of a single endothelial cell layer. We're going to come to that in a minute. And that's where the oxygen is going to diffuse into the blood. Then that oxygenated blood from the pulmonary capillary beds will be gathered together and join the structure I showed up above, and we'll get to it in a minute here, will be the pulmonary veins that will be carrying blood from the lung, pulmonary, back to the heart. Toward the heart is a vein, and that's the circuit. Okay? Yes?
So when the blood leaves the pulmonary artery, it goes to the lungs and capillaries?
So we haven't gotten to it yet, but we're going to spend a lot of time showing that there is this pathway that goes arteries to arterioles to capillaries to venules to veins and back to the heart. So that will be the sequence of events. This is a sequence of tubes. Now it's worth pointing out here, and we're going to emphasize it later, this is a closed system. The blood never leaves these tubes. Things are working right. The blood stays in the tubes, and they go from tube to tube to tube. Now, the capillaries, these capillaries, put in green, the capillaries are the exchange vessels. And these capillaries, if you look at them closely, are composed of nothing more than a layer of endothelial cells. Hopefully before we leave today, we'll see that these endothelial cells, a single layer of endothelial cells is continuous through the cardiovascular system. Doesn't matter whether you're talking about arteries, arterioles, capillaries, venules, or veins, there's a continuous layer of endothelial cells that extend into the chambers of the heart and across the valves. But first, let's talk about the other side. Over here, this is the left atrium. As we saw on the right, we have atrial myocardium. So the open chamber of the atrium is surrounded by this muscle, the atrial myocardium. The tube coming in here, I wish I had not put these out. The tube coming in here, of course, is the pulmonary vein. The pulmonary vein, it tells you it's carrying blood toward the heart. This pulmonary vein, because this is blood that's just left the lungs, this is going to be oxygen rich. As we saw in the 4-box model, the blood flows into the atrial chamber. Between the atrial chamber and the ventricular chamber, of course, is that atrioventricular valve. So again, this is the ventricular myocardium. And here, this open space, as though we've cut the heart open, this would be the ventricular. I should say left ventricular chamber. So pretty much the same system, left and right. It's one where we have two pumps. A right pump dedicated to moving blood to the pulmonary vasculature, and a left pump dedicated to moving blood to the rest of the vasculature. The tube out of the left ventricular chamber is this structure called the aorta. And it has a valve here called the aortic valve. Well, just as we showed above, we can imagine these valves as being doors. And again, this valve between the atrium and the ventricle, this is a fairly floppy valve. So we're going to see this a little later, we're going to see is a low pressure return. This is not high pressure. After the blood's moved through all those capillaries in the lung and moved through all the capillaries in the systemic vasculature over here, this is low pressure. That pressure opens this atrioventricular valve on both sides. The low pressure now moves the blood into the open chamber. What we're going to see is now, this is muscle. This muscle is going to contract. And when this muscle contracts, it increases the pressure. When it increases the pressure on the fluid inside the chamber, that slams this valve shut. They're floppy. This valve out here, though, is pretty stiff. It's going to take a lot of pressure to open. It's nothing fancy. It's just a stiff door. And what you have to do is you have to increase the pressure in here. The pressure in the chamber, I'll say P sub C, has to be greater than P sub A, which is the pressure on the far side of the aortic valve. Now, when you leave here today, this is worth noting today, when you walk out of this building, inside the building, is at negative pressure. So there's greater pressure in the outside of the building than there is in the inside of the building. And I've done this 1,000 times. I go to the door out here, I hit that panic bar, and I think I'm just going to push it open. And I start to put my arms into it, nothing happens. And then finally I have to get my legs into it and finally force it open. And the reason that it takes so much force to open that door is because the greater pressure on the outside, on the inside. You have to put more pressure on the inside of that door to open it than the air pressure is on the outside. We'll see that's the same story here. We have to generate more pressure on this chamber side of the valve than we have pressure on the aortic side of the valve to open it. And then when the pressure is greater on the aortic side than on the chamber side, that valve will swing shut. These are just doors. Doors responding to the pressure in either the tubes or in the chamber. What we'll do next time is we're going to lay out a series of cells in here called the endothelium. Now I'll let you all go, but there was a question.
The drawings, are they anatomically accurate with where the valves are at?
They're anatomically accurate to where the valves are in terms of between the chambers.
Where the pulmonary artery goes out?